given to you by Dr. DeCenzo from ETS Labs. He often would come and give a guest lecture in addition to showing up to our lab to conduct the qPCR Scorpions lab. So I didn't want you to miss out on it because it is important. And so I'm going to present these slides to you on minimizing the risk of problematic fermentations. So this slide just shows you an overview of fermentation and looking at the normal progression from a sluggish to a stuck fermentation. So again, sluggish being the rate is slow, but it's still progressing, and then obviously stuck is what it is. The fermentation is stopped. Fermentation rate depends on a lot of things, but these are the main ones. Total viable biomass. So again, that's a reference to the amount of viable yeast cells that are conducting the primary fermentation. The rate of sugar utilization by individual cells. So it's not just enough to have a bunch of viable cells, but you've got to have cells that are optimally using the sugars. And that's why the third bullet point is important. Viability is not the same as vitality. And so this little slide just illustrates that dichotomy, viability versus vitality, where you have viability on the left, this Grandpa Joe character. These are not my slides, by the way. This is Rich DeCenzo, so I don't know who the hell Grandpa Joe is. Anyway, you have Grandpa Joe on the left, and then Usain Bolt on the right, showing you that they're both viable, but there is a huge difference in vitality. And so one of the ways we measure viability versus vitality is, two of the ways we measure viability versus vitality is on the left. You can see the use of methylene blue staining and a hemocytometer, which I talk about in the lab portion. And there's also slides for this on the tools used in our field, the field of wine micro. And so you're looking at a methylene blue stain of live and dead cells. The dead cells are blue in this particular image, as you can see here, and all around. And then you can see the small squares of the hemocytometer that you can focus in on and make two quantifications. You can total quantification and live versus dead cell. And again, these calculations and how to do this are in the lab manual. There's videos posted about it, and there's also a specific lecture about it in the tools used in our field packet. Over here is another approach for measuring viability and vitality. CO2 production or change in sugar concentration. Typically you can use refractometer or hydrometer to generate this, or you can measure CO2 directly using a CO2 meter. One of the things that ETS Labs likes to use is a ViCell XR counter. And so if you get a chance, maybe you can do a quick Google search or something to look up the ViCell and how it's used. But it's basically a similar chemistry as this. They're using a dye that can penetrate dead cells but not living cells. So it's a similar concept. But again, you can look that up on Google. Metabolic basis of reduced sugar utilization is primarily membrane transport capacity and specifically decreased sugar uptake. And typically those are HEXOSE transporters, these transporters in the membrane that can take in 6-carbon sugars. And so these are, again, proteins that shuttle sugars across the membrane. So I have a cartoon of that coming up. But the HEXOSE transporters, they're a pretty big family, multi-gene family of transporters that yeast cells have. There's 18 members in the HXT family, and again, HXT for HEXOSE transport family within Saccharomyces. They're typically downregulated by intracellular glucose, so that should make sense, right? If the cell is flush with glucose on the inside, then they don't need more transporters, and so they may shut some of them down so that they're not working, or they may also stop gene regulation to produce more of them. But they're also upregulated by extracellular glucose. And so, again, if extracellular glucose is high and intracellular glucose is low, that would trigger an upregulation where the cell would actually favor production of more of these regulators. But again, if intracellular levels are high and extracellular levels are high, the yeast cells will settle on a sweet spot where they can get the most activity without choking themselves to death. So transport begins when a sugar attaches to a specific binding site on the HEXOSE transporter, which then causes a conformational change in the transporter to allow it to come in. And so that this cartoon sort of illustrates the concept where you see the yeast cell membrane and then the inside world of the yeast, the cytoplasm, and then this hexose, a 6-carbon sugar that would then be moved by this hexose transporter here into the cell. The role of the cell membrane is integral, obviously, because cell membrane fluidity is critical for sugar transporting. If there's not enough fluidity, then transport doesn't take place, and that can happen if it's too cold, for example, and the membrane starts to solidify. And conversely, if the membrane's too fluid, and this often happens when it's too warm, and you get and again, it shouldn't surprise you when a fatty material like butter, for example, is put in the refrigerator or the freezer, it turns pretty hard. And obviously, if you leave it out on a hot summer day, it gets really loose. The most important thing is you can't go back. Once cells reduce their sugar consumption rate, then it's very difficult to resuscitate them. By the time you recognize that there's a reduced rate, it's usually too late. And at this state, in my experience, usually that means yeast viability starts to drop below 50%.